Hello viewers. Topic of discussion of this video is on a fungal disease caused on a very important plant, that is coffee. The name of the disease is coffee leaf rust, which is commonly called as CLR. Coffee is the most important agricultural commodity. It provides important export revenues and labor opportunities for many tropical countries. In the Indian context, coffee growing started with an Indian Muslim saint by name Baba Budan, who, while returning from a pilgrimage to Mecca, brought 7 coffee beans from Yemen to Karnataka. Hence, Arabica became more popular. Coffee production in India is dominated in hill tracts of South Indian states, with Karnataka accounting for 71%, followed by Kerala and Tamil Nadu. Indian coffee is said to be the finest coffee grown in the shade rather than the direct sunlight anywhere in the world. The two well-known species of the coffee grown in India are Arabica and Robusta. These two are marketed under the brand names Arabica and Robusta. Contents of this video: introduction, disease symptoms, etiology and infection process, disease cycle, and lastly, disease management. What is coffee leaf rust? Coffee rust is induced by a fungus Hemilia vastatrix. It is a major disease of Coffea arabica, which constitutes about 75% of total coffee production. The fungus manifests as yellow spots that first affects the undersides of the leaves and then develops into a yellow-orange powdery dust that is easily transferred among plants. Coffee leaf rust can be traced back to the birthplace of coffee, that is Ethiopia. From here it followed coffee production around the world. In the late 19th century, Sri Lanka was one of the world's largest coffee producers, exporting over 100 million pounds each year. But in 1892, a leaf rust epidemic wiped out all of the island's coffee trees, and producers changed to growing tea. Coffee leaf rust spread to India from Sri Lanka during 1870s and caused large-scale damage to Arabica, leading to panic among the growers. In 2012, coffee leaf rust became epidemic. There was a major increase in coffee rust across 10 Latin American and Caribbean countries. So CLR has become one of the most well-known diseases in the history of plant pathology. Symptoms of the disease. The symptoms of coffee rust include small yellowish oily spots on the upper surface of the leaf, which expand into larger round spots that turn bright orange to red and finally brown with a yellow border. As these spots gradually increase in diameter, the rust pustules appear as orange-yellow on the lower surface with masses of orange iridospores. The lesions can develop anywhere on the leaf. They tend to be concentrated around the margins where dew and rain droplets collect. The centers of spots eventually dry and turn brown, while the margins of the lesions continue to expand and produce iridospores. Premature leaf fall. That is, the infected leaves drop prematurely, leaving long expanses or twigs devoid of leaves. So now the etiology and infection process. Hemilia vastatrix is an obligate parasite and a hemicyclic fungus. It produces uredinospores, teleospores, and basidiospores, whereas the pycnospores and esiospores are not known. Uredinospores and teleospores are produced in the same source but at different times. Uredinospores are dikaryotic and represent the asexual cycle, reinfecting the leaves whenever environmental conditions are favorable. Infection process. The infection process involves the growth of uridospore from inoculation until lesion formation. It involves specific events. First one is germination. Uridospores germinate only in presence of free water. It often produces 2 to 3 germ tubes simultaneously. The germ tubes either directly or after branching produces aprisoria on stomata. Penetration. On the dorsal surface, the aprisorium produces a vesicle that is a cytoplasm sink. This is in turn penetrates the stomata by means of an infection hypha and thus gains entry into the substomatal cavity. Diagram A, aprisorium over stomata and intercellular hypha with an haustorium, whereas in diagram B one can see oppressorium over stomata and intercellular hypha with an haustorium. AP stands for appressorium and H for haustoria. The next step is colonization. The infection hypha ramifies intercellularly in the substomatal cavity and nearby tissues and penetrate the cells by means of haustoria. In diagram A, intracellular hyphae with epidermal and mesophyll cells are visible. In B, haustorium with spongy parenchyma cells can be seen. In C, intracellular hyphae in the spongy parenchyma are visible. Now, the sporulation process. The fungus sporulates through the stomata rather than breaking through the epidermis. The mycelium sends out stalks through stomata which bear the uridospores. The uridospores are reniform in shape. Diagram A, uridinosaurus protruding through the stomata, and diagram B, uridinosaurus with uridinospores. Dissemination process. The dissemination process in Hemilia vastatrix consists of three sub-processes from the mature spore to the deposited spore stage, like spore liberation, dispersal of spores, and deposition of spores on leaf surface. The uridinospores are mainly wind disseminated. The rain also plays a major role in the dispersal of spores. Indeed, it was once considered the most important agent. Uredospores after their release from Uredosaurus. Germinated uredospore with germ tube and aprosorium over stomata on the lower surface of the leaf. Intracellular hyphae within the epidermal as well as mesophyll cells with haustorium. Lesions on the leaf. Uredosaurus protruding through the stomata and release of uredospores from the sorus. Disease management. Factors that affect the severity of rust disease includes amount of rainfall, number of infected leaves at the beginning of an epidemic to supply the spores, that is the inoculum, and the amount of foliage present on the trees. The first method is avoiding shade. Excessive shade increases rust disease as it keeps leaves moist for longer period, and this also increases the number of spores that germinate. Pruning the branches to allow more air circulation can dry the moisture on the leaves and prevents the pathogen from infection. The second method is providing proper tree spacing. If the trees are too close, then the disease develops at a faster rate because the leaves remain moist for longer time, and this increases the number of spores that germinate. Hence, proper spacing must be provided while planting trees. The third method is growing disease-resistant varieties. Robusta coffee varieties are generally considered as resistant to all races of coffee leaf rust. Natural hybrids between Arabica and Robusta are available, and hence these varieties should be used. Use of fungicides. Copper containing fungicides or copper sprays can be applied early in the season, but fungicides must be present on the leaves before infection occurs. The disadvantage is that copper accumulates in the soil. It can reach toxic levels and affect both organisms as well as the environment. Therefore, proper care should be taken. This is a photograph of a healthy coffee plant with ripe cherries. Another one, coffee cherries unable to ripen because of coffee leaf rust. Thank you for watching.